Okay then. Hello everybody. Welcome to this edition of our Institute live stream series. Today I will be gladly speaking to you about a very interesting topic in biology. I was summoned here to speak about what I myself like to title the Amazing Apparatus. These are the insect antennae. So before we begin, I'd like to introduce myself. My name is Sin, and also I'd like to introduce you to the very fabulous, small-scale, yet very far-reaching world of the insects, and of course their most amazing appendage, the antenna. So, as we begin, I'd like to give you a little bit of a taste of the exact diversity of insects, and also the diversity of the antennae that they have. As you can see on this picture, insects come in a very, very wide variety of shapes and sizes and colors. And on the right, you can see that even their antennae tend to be very, very diverse in morphology between different species and different clades within the insects. All insects, in fact, or maybe not all insects, possess two eyes and two antennae, and that's more or less a defining feature of the insects. And these insects come in various different groups, which we have grouped together. And in the field of research that we concern ourselves with when we speak about insect antennae, tend to encompass the following insects. Of course, it can get broader. So today I'll be mainly focusing on dipterans, on the true flies, because they tend to be the most best studied insects among all of them. And they of course feature the very prominently studied model organism Drosophila melanogaster, the vinegar fly, as you can see here on the left. Of course, there are other insects that we tend to study when it comes to the neurobiology of the sense of smell and the sensory biology of antennae. These include, just to give you a few examples, the lepidopterans, so the moth and the butterflies, and of course the hymenopterans, the big group of insects which includes a lot of eusocial creatures such as the ants, the termites, wasps, and bees. And of course, we would all be amiss if we didn't mention the beetles themselves, which are a very fascinating clade. They include a quarter of all animal species, and even within the insects, they include they make up 40% of all insect species themselves. So they should not go unmentioned. So, why are insects important to us? Well, of course, they're important to us for two main reasons. One is because they are such a very vital and essential part of the ecosystem in their functions as maintaining the entire ecosystem, and of course also in cycling nutrients and as being part of the food web. So they perform a lot of different things, as you can imagine. For instance, the beetles, the most numerous of species that we have among insects, they are very important scavengers, and they cycle very many nutrients from the top ground back down to the soil. And of course, they're included in a lot of different functions in the environment, such as aerating the soil and so on. But of course, insects are not only interesting because of their ecological commitments, but also because they're very interesting to us as humans in our own human endeavors. So they actually make up a very important function as pollinators, and they also fulfill a very important role as pests, and they are of course very interesting to us because of that reason. Likewise, they also function as biocontrol agents, and they have actually been applied as biocontrol agents. So we have actually employed and deployed several insect species in order to fight other crop pests and so on. And of course, perhaps one of the most notorious interests for humans in terms of insects is regarding mosquitoes and their ability to function as disease vectors. They are of course not the only insects that can act as disease vectors, but we study them intently to be able to call the spread of many diseases. So these are actually the two main reasons why insects are fascinating to us. and to biologists in general. So today I've broken up my talk into 5 chapters, into 5 sections, and I'd like to go through them one by one to give you a little taste of exactly what it is that it is when we talk about insect antennae. So the first thing I'd like to discuss is why antennae exist in the first place. Why are they there? Why did they evolve? And why do they take on so many different fabulous features among the insects? Then I'll move on to discussing the arsenal of genes and proteins that are actually expressed and that are under pressures of evolution within these insect bodies, and how they actually contribute to being the forefront and the interface between the outside world and the inside world of the insect. Then, of course, I'll move on to the structure of the antenna and how all these genes and proteins actually coexist, and how they are co-expressed and built into these antennas, and how these antennas actually function on a cellular level and on a tissue level. From then on, I'll just like to very briefly mention a couple of levels of biological complexity that we have to contend with when we speak about antennae. And I'll try to finish up the talk by giving you a little primer on how we explore the antenna in the lab and all the different methodologies we have in place in order to study these little fascinating appendages on these insects. So without further ado, I would like to pose the question, why do antennae exist? Why do they even exist in the first place? The answer is actually rather simple on the face of it. Antennae exist because they are a sensory apparatus that detects the world around them. And the world around them is very, very complex, and it's brimful with useful signals, both chemical and auditory. And these signals are intermixed and occur in a very, very complicated fashion around the world, regardless of the environment. And of course, these signals need to be integrated and detected correctly enough for these insects to be able to thrive, reproduce, and survive at an optimal pace so that they do not face extinction. It's a very, very simple explanation. So whenever the insects think about the wider world and they detect everything, they must use these signals in order to make decisions. These decisions come in very, very many different flavors. I will just give you a very short list of all the potential things that these flies must integrate and use. So for instance, flies are tasked with mate selection in their ability to detect the natural environment. They must be able to find mates, discriminate among the mates, and pick the best mates in order to sexually reproduce. And of course, they must also be sensitive to courting rituals and all the different signals that come with courting rituals. Of course, the flies must also forage, typically for food, also sometimes for nuptial gifts in the actual courting processes. They must, of course, also find suitable locations for laying their eggs on substrates that are definitely not going to harm the offspring and that have to be suitable for the offspring to have the best chance of actually making it into adulthood and thriving. The flies must also decide on when to perform social behaviors. So they must actually detect the outside world in order to be able to aggregate, in order to know when to be aggressive, when to be territorial, and to perform other social behaviors. There's also, of course, little things such as larval avoidance of cannibalism. So you can imagine if a bunch of siblings get laid on a substrate by a mother, they must not be eating each other cannibally, and they must avoid eating each other. So the whole world really presents them with a lot of different complicated challenges that they must make sound decisions on. Of course, there are plenty and plenty of more examples, such as nest and hive navigation. So this is, for example, with ants. They must be able to walk to a source of food, back then again to the nest, and communicate this to the colony. There's also, for example, instances of predator avoidance. So, for example, many insects need to avoid parasitoids from using them as a host. And of course, there are very many insects which are carnivorous, such as the dragonfly, which is very insectivorous. It's a very good hunter. And of course, the very famous praying mantis. And it might also be a good example to mention the mosquitoes, who also must perform host seeking and find suitable hosts from which they can actually draw blood and sustain themselves and their offspring. So in this sense, the antenna itself is a barrier, and it's a vanguard between the actual processing in the brain and the chemical world that you find around them. So this is a very important sensory gateway, which has its own encoding range, and it has its own input limitations, which we try to understand in order to be able to first describe and understand why these insects behave as they do, and also what kinds of things we can manipulate and exploit in order to be able to better put these insects to perhaps an applied task. I should mention before we proceed that the antenna itself doesn't only perform chemosensation. Of course, today I'll be focusing mostly on the chemosensory aspect of the antenna, so the olfactory side of things and a little bit about the gustatory side of things, so the taste and the sense of smell. But it should be very clearly noted that the antenna itself performs a variety of other functions which are extremely important to the survival of the fly, such as heat sensing. They perform thermosensation on the antenna. Gravity sensing, so knowing which way is up, which way is down. They also perform moisture detection, so being able to detect how humid an environment is. And of course, a very, very important sense of hearing is also housed in the antenna. Of course, not all insects use their antenna as their primary hearing organ, but at least in a lot of flies and other insects, they use the antenna as an auditory organ. Just as a brief aside, this auditory sensory system is housed in the antenna in the second segment, which is the one here, as you can see. And it hosts a little organ called the Johnston's organ, which is sensitive to stretching and pulling, which is caused by the rotation of the third segment, this segment as you can see here. In this way, the flies, when a sound is presented to them, they can accurately judge what the frequency of the sound is and what the intensity is of the sound based on how much the antenna is rotated and deflected by the sound waves, and how much this activates the neurons that are connected to this little joint, this rotational joint inside. So, of course, the sense of hearing is very important to flies because they actually use a lot of singing in their courtship process. So they have a very elaborate mating dance and process that they have to go through before they actually attempt copulation. So as you can see in this figure, they have to first orient to each other, they have to tap on each other, they tend to sing very specific songs, which tend to be specific also to the exact species. You can see here, for example, that Melanogaster has a very different song than Simulans in terms of the sine pulses that they provide, the gusts of wind that they can create, and also the intervals between these. So hearing is a very important part in the copulation process and in the courtship process for flies. So of course the antenna is also a very central organ with respect to the sexual reproduction of flies and of course other insects. However, I digress a little bit. So there is a question. The insect sense of hearing is more or less straightforward. The antenna rotates based on sound intensity and pitch. But now, what about the sense of smell, the other grand function of the antenna? What about the sense of smell? Is it that straightforward? I'd like to pose to you the fact that it's not really as straightforward as hearing for a lot of different reasons. And the antenna has to contend with very different features that we have with odor space as opposed to, for example, visual space, which might be, for example, dependent on wavelength and intensity, and sound space, which might be dependent on the frequency of a sound and its intensity. There are quite a bit more dimensions at play here. So the first problem that we have is stimulus dimensionality. we have a vast array of very different molecules that come in all different arrangements and combinations. And there is no one feature, no one array that you can arrange these molecules by. And these actually have to be interpreted by the animal and by the antenna. And this is a very difficult problem. And the antenna doesn't really solve it in simple ways, let's say categorizing everything into acids or categorizing everything into esters or aldehydes. It is a very difficult problem for an olfactory system to evolve to be able to detect and discriminate between all these different kinds of molecules. As I already mentioned, there are also potentially infinite numbers of informative molecules or potentially informative molecules, and the combinations thereof, so mixtures, that are of course present in any environment and in any habitat that you can provide to the insect. So with flies, we, for example, understand that most of the molecules and odorants that are available to the fly for processing are very small molecules on the order of the size of tens to hundreds of Daltons in size. These molecules tend to be very volatile, so they have a high vapor pressure, and they also, of course, tend to be fat-soluble and organic. However, there's still a very, very, very large spectrum of molecules that have to be discriminated by a sensory organ such as the antenna. Another feature of odorants and smells in general is that they are noisy. They come at very unpredictable times. They come in mixtures. They come in very widely dynamic ways. So they come in very high concentrations and low concentrations, intermittent concentrations, and of course they fluctuate in time. So these are all problems that the antenna and any sensory system devoted to olfaction must contend with. So now that we've tackled why antennae exist, let's dive into the olfactory arsenal, the actual genes and proteins that are in place that interface with the outside world, with all these molecules that we've described, and transduce this information into the organism, into the body. So, simply put, the entire arsenal that we have discovered so far, we call this the Chemosensory Arsenal, and this is basically a repertoire of receptors and other molecules. First, I'll begin by describing some of the receptors and all the different families of receptors that we have. So the most well-studied of the families are the olfactory receptors. These are 7 transmembrane domain membranous proteins that sit on the membrane of neurons. And there are very, very many of them in insects. So they are sometimes present in dozens and dozens of variants, or sometimes in hundreds of variants across different insect species. And typically each receptor responds to one or very few odorants. And typically there are only one or few receptors that sit on the membrane of the neurons, determining their type. So of course, these receptors actually interface and bind ligands from the outside that have been dissolved into the organism, and they typically tend to produce behavioral outputs that of course depend on the higher brain processing that later follows once the nerves send out a signal back to the brain. There are a couple other very important receptors that I should mention among this repertoire. We have, for instance, the gustatory receptors, here shown in blue, the ionotropic receptors, which function in a very different way, although with very similar principles. And of course, there are also other receptors such as the PPKs and the TRPs, which I'll not go into. They also constitute a part of this repertoire of receptors that is in charge of interfacing with the outside molecules. It would be amiss not to also mention the other part of the arsenal, which includes auxiliary proteins, which are actually not responsible for directly, shall we say, transducing the information from the odorant into the body, but are very, very important for actually capturing molecules and interacting with molecules together with the olfactory receptors so that a complete response can occur. So among these auxiliary proteins, we have SNMPs, which sometimes sit which sit on the membrane along with other olfactory receptors. and the very important OBPs and CSPs. So these are the olfactory binding proteins and chemosensory proteins, which tend to be secreted into the environment where they act as helpers to the receptor repertoire for interacting with all these different various chemicals. I'll get into them very shortly, but you must understand one of the main principles to take away here is that different insects have different repertoires, and they may sometimes share orthologous receptors between each other, but each different insect species tends to have a slightly different repertoire, and this actually grants them the ability to detect some chemicals while other chemicals might remain invisible to the insect. And of course, every insect might have very different needs and very different requirements for which smells might be important for it, therefore creating a kind of distinction in repertoire between the different species that we have to contend with here. So, what is this giant repertoire good for? How do the receptors actually make the neurons fire signals into the brain? Here, I'll try to very, very briefly describe how the receptors sit on the membranes and how they transduce the information. Please don't be intimidated by all of these diagrams. They're just there as guides for us. So typically with these receptors, each receptor that might be receiving a ligand, a chemical from the outside world, is present with a co-receptor that is shared between different receptors among different neurons. So in the case of ORs, we have different receptors for different chemicals, and these sit together with a common co-receptor called ORCO, or olfactory receptor co-receptor, and they function together in order to form channels through which ions can flow and therefore depolarize neurons. These olfactory receptors and gustatory receptors and ionotropic receptors share no similarity, no sequence similarity with vertebrate, nor other invertebrates, such as C. elegans, receptors, which are typically involved in olfaction, such as the G protein coupled receptors. The membrane topology is very distinct and very different. So, especially if you look at the olfactory receptors, the C terminus is facing the outside, and this is not the case typically with G protein coupled receptors, where the N terminus is facing the outside. Now, these complexes that tend to form between receptors and their co-receptors function as ligand-gated ion channels, but they can function completely independently of the G protein coupling that tends to be present on the inner side. And of course, these receptors sometimes function in tandem with other proteins that I mentioned, such as SNMP1, which was one of these auxiliary proteins that I mentioned earlier. There are basically two important features of how the transduction occurs. One of them is the direct and very fast ionotropic response that is allowed for by the formation of these ion channels that are gated. So these grant the insect speed and quickness in being able to receive a signal from the outside. So you can imagine when an odorant arrives, it can bind to a binding pocket on the receptors, and this immediately causes the cell to know about the odorant from the outside by allowing an ion current to flow into the cell. So this grants olfactory receptor speed, which are very important for the animal. However, there's also a second part, which is very important. We call this the metabotropic side of the response, and this is the slower, amplifying, gain controlling response that occurs inside the cell, which grants the transduction process an ability to control and increase its dynamic range and to increase its sensitivity to even sub-threshold stimulations of odorants. So even if you were to present these receptor complexes with very, very small amounts of an odorant, the cell within could still be able to detect these and amplify and adapt its response based on these things due to the metabotropic side of things. Of course, I will not go into any of the detail, but the most important two key takeaway things from here are that we have a very fast response and also a slow response that is governed by the receptors and the cellular processes within. So, now that we've spoken about the fact that these receptors allow for very, very quick transductions and transductions over a very large range of low concentrations and high concentrations, do all the receptors respond to one odor? Do single receptors respond to all the odors? How does this really work? What about odor selectivity? So this is also another principle that I would like to share with you, and it's a very important principle when speaking about the sensory system of insects. The receptors are tuned to a range of chemicals, and this depends on the receptor that we're talking about specifically. So it depends on which receptor within the repertoire we're talking about. We have, for example, receptors such as OR69A, which includes an ability to respond to very, very many different ligands that you present it with. Very many different odorants activate this olfactory receptor. However, this can also be contrasted with other olfactory receptors such as OR67D, which actually responds only to a single odorant and will discriminate against all other odorants and not respond to any of them except for for one odorant that it is responsible for. We have actually mapped out the tuning curves, so to speak, of all these receptors, and we can see kind of a very nice spectrum of some that respond to very, very few ligands that are very, very selective for what they bind and what they detect, and others that are not very discriminating and that will bind very, very many different odorants. This, in fact, this entire principle leads to combinatorial coding in the insect fly, because the actual fly might not be able to discriminate between mixtures perfectly, but this is a task that is relegated then to the higher brain centers and to the antennal lobe in order to actually discriminate and sort out between what the actual antenna overall is sensing. So here I've labeled a few of the olfactory receptors that are incredibly specific to one or a maximum of 2 or 3 ligands. We have, for instance, OR67D, which responds to a pheromone called CVA in flies. This receptor knows no other binding partners. We have also OR56A, which binds to a very important and ecologically relevant odorant called geosmin, which is secreted by bacteria. So this is a very important receptor dedicated to a very narrow pathway that informs the fly of bacterial presence. And of course, another receptor, OR47B, which responds also to a very narrow subset of pheromones. As you can tell, these are very important functions in order to actually smell potential mates and to be able to smell pheromones on each other, and also to be able to smell noxious and toxic microbes around. So these receptors have evolved very narrow tuning curves, while others respond to very many different odorants. I did say I would return to the odorant binding proteins and the chemosensory proteins very briefly. These are the accessory proteins that I spoke about that also occur within this repertoire, and these proteins are found all across the insect bodies. Typically they are found in antennae and mouth organs and other chemosensory structures, and these interact with odorants by solubilizing them, by capturing them and releasing them in various different ways, and controlling the actual kinetics of how these molecules become presented to the sensory system. All of these OBPs and CSPs have been, or not all of them, I should say, many of them have been studied, and there are a few key features that we understand about them. They, for example, become transferred during mating because they tend to carry pheromones in very many species. Very often they have nothing to do with sensory systems, and they are involved in processes such as development, regeneration after injury in, for example, cockroaches, the maturation, for example, of honeybee eggs, in pupation, in ecdysis, and of course also in phase shifting, for example, in locusts. These molecules tend to be very important for carrying very important hormones and fulfill this function very similarly to molecular chaperones that might, for example, be extracellular. And they are very reminiscent of the Holdes function of chaperones in their ability to sequester and carry and release molecules around the body. But of course, these OBPs can also capture dietary nutrients that might be very difficult to absorb. They also have an anti-inflammatory action in blood-feeding insects, which is very important for them. And without these binding proteins, these insects would not be able to perform their functions very well. And also, of course, these molecules have been implicated in insecticide resistance. So they are a very fertile ground for research. And as I mentioned, that they also have made an appearance in applied sciences as sensitive biosensors. They have also been applied as pollutant scavengers, and of course, even in the perfume industry as controlled releasers of fragrances. Very successfully, I might add. Anyway, now that we've described kind of the backbone of the repertoire of genes and proteins that actually exist and carry out the function at the periphery, now we're going to delve into the antennal structure and how everything is organized at the cellular and at the tissue level. So here, as you can see, we have scanning electromicrograph of the fly head. And as you can see, these are the antennae that we've been talking about so far, which are divided into a first segment, which is barely visible, a larger second segment, which houses the hearing system, and the third segment and the arista, the fourth segment, which is very important in chemosensation. This may perhaps be a little better picture to illustrate which parts are involved in olfaction. And in the sense of taste, we have the third segment of the antenna, and we also have the maxillary palps, which fulfill this function. And as you can see from these pictures, one very important and obvious feature of these organs is that they are very hairy. And these hairs are called sensilla, as you can see here on these close-up pictures. So, as I already mentioned, these insects have two antennae, and especially with regard to Drosophila melanogaster, our most studied model organism, we find that between the two antennae, it has about 820 sensilla, which are these little hairs. And within these sensilla, we actually have innervating neurons, which are doing all the actual transduction process from the outside to the inside. These 820 sensilla house about 2,400 of them. And within these, we actually have the repertoire of receptors being expressed inside these little hairs. There are, of course, two antennal features that I want to not forget to mention. We have the sacculus, which is a three-chambered pit that lies somewhere here, and it's very difficult to see, which houses a lot of acid-sensing ionotropic receptors, especially. They can also detect amines and ammonia. And of course, the arista, which is this large hair-like structure that protrudes. Very often it's called the fourth segment, which also houses neurons, important for thermosensation, and itself is used to actually capture the vibrations in the air that allows the second segment to hear, basically, for the fly. So this is what the antenna looks like from the outside. It's covered in a lot of these hairs. The hairs themselves are not all created equal. They're very morphologically distinct from one another. We have, for instance, 3 or 4 different classes of sensilla that cover and pepper the antenna. We have trichoids, which are very long hair-like sensilla. We have basiconics, which are club-shaped sensilla. And we have these kinds of pegs, kind of invaginations within the cuticle. And these are the coeloconic sensilla. Of course, there also are different other hairs on the antenna itself, which are called spinules or extrasensory sensilla. But the key takeaway here is that the antenna itself is not covered with just a single type of sensilla, but they actually come in very different shapes and sizes. The sensillum itself is a very versatile and conserved sensory organ and does not necessarily only appear on the antenna, but also appears throughout the body and on the wings and on the legs and on the abdomen of the fly. These sensilla are very, very well conserved sensory organs, and they are featured in different sensory modalities, such as mechanoreception and the like. And they are also very homologous to each other, so they always built up in a very similar fashion. They're also very complex, and all the cells are retained upon the formation, and all cells are thought to contribute to the final ability of each sensilla to carry out its sensory functions. So as you can see here, this is what it looks like during development when you have an epidermis. It differentiates into these kinds of single units, which then form entire sensilla from themselves. And this happens thousands, if not millions of times across the insect body. And this is the beginnings of how these sensory organs are formed. So as I already mentioned, they come in very different sensory modalities. They're involved in the Johnston's organ, in hearing, in this kind of arrangement. The sensilla also appear in chemosensation, which is what we'll be mostly focusing on in this arrangement, and also in mechanoreception around the whole body. Very important for the insects to know exactly where their bodies are being stretched, and they also occur in joints and on the surface of the body as touch receptors. So I'll go into a little bit detail about exactly how these complex units look like. For olfaction, they look like this. Of course, it's a little bit of a cartoon, but I think it's a little bit of a more complete illustration of how each of these hairs looks like. So I'll try to explain now, one by one, what all the different features are on this sensillum in order to just give you a better understanding of all the different things that come together in order to form just a single hair on the outside. First of all, we have the epithelial structures. We have the hard chitinous cuticle that is on the outside, which is lined with pores that allow access for odorants, for example, to enter into the hair itself, into the sensillum. We have the basement membrane here colored in yellow. We have a lot of epithelial cells that surround the system. And a very important feature is the lymph. So anytime that any odorant wants to enter into this sensillum, for the fly to detect anything, it must actually first be dissolved inside an aqueous lymph that here I've color-coded in bright yellow. Then we have 3 or 4 different kinds of supporting cells, here shown in green, blue, and purple, that fulfill a lot of secretory functions and that are thought to degrade and clean up and maintain the lymph itself. And of course, in aiding the neuron in various ways, especially like in glial ways. So we have glial cells that are present in the system, and also the innermost cell, which is called the thecogen cell, which is poorly understood, but has thought to function in some way as an ensheathing cell, similar to the glial cells. So between the epithelial structures and the supporting cells, in the very most core of every sensillum, you have one, or in some cases few, olfactory sensory neurons actually innervating the hair itself. And this is the true star player in the system. This is where all the olfactory receptors sit, and this is the cell responsible for actually communicating with the outside world. I'd like to just make a little bit of a aside and bring our friendly coronavirus into the discussion. As you have seen here, there are very many cells, and usually we focus on the olfactory sensory neuron because of its receptors and because it's actually interfacing with the odorants itself. But you may have heard that getting coronavirus, and also, of course, a lot of other different illnesses, can sometimes induce anosmia, which is just a reduced ability to smell. And in the specific sense, in the specific case of coronavirus, we now understand that the coronavirus does not actually infect the olfactory sensory neurons in mammals, but actually the supporting cells that are found around. So this is actually not in insects anymore. We're just talking about mammals here. But I thought it was a very nice aside to mention that COVID actually infects the supporting cells, and through some unknown mechanism, there have been several hypothesized, actually leads to a failing of your ability to smell things. So this actually a little bit shows you that the supporting cells can also be very important players in the entire system. So it is not only just the neurons themselves that do the work, but it is actually a complete unit together with the supporting cells and the lymph in which they are bathed that actually performs all the processes. So just to give you a quick recap, the antenna looks like this. It has a first, second, and third segment with the Johnston's organ in the second, which we will omit for now. And the third segment of the antenna is completely lined with very many different hairs. And these hairs are innervated by neurons, and these neurons, in a very, very stereotyped fashion, innervate specifically different classes of them, and the whole repertoire of olfactory receptors is segregated into different types of neurons which sit within these sensilla. And this kind of hopefully gives you a better picture of how the actual sensory organ performs its olfactory functions. So just to very briefly recap the antenna, as I mentioned, densely populated with hairs called sensilla. There are different kinds of sensilla, and each sensillum is a site, isolated site of a chemosensory unit, which includes the lymph, the neuron, and the cells. And together they form the basis upon which odorants become transduced into neurological signals for the brain by the olfactory receptor neurons. So there are several levels of biological complexity that we have to contend with. I would just like to give you a very brief primer on all the different things we have to think about when we think about the antenna. That it's not a simple story. Of course, in biology we have very many different scales upon which we can appreciate a biological process. So we, for example, talked about the molecular side of things with the receptors, the cellular and tissue and organ side of things with how actually the cells come together and how they're arranged in order to best transduce these chemical signals back to the brain. And of course, the higher levels of the ecosystem and how the organism behaves are very important. So we have very many different, let's say, features of a sensory system that we have to understand to better understand the entire process. And all sensory cells have these 2 or 3 features in common with each other. They tend to be adaptive, so that if you present a neuron, an olfactory receptor neuron, with a very high amount of a stimulus, that it adapts and it decreases its firing, and not that it's always constitutively firing at maximum speed. And also an ability to sensitize, so that if you provide it with a sub-threshold stimulation, that it can actually utilize this and become more sensitive over time if the stimulus is present in very, very low amounts or in very, very intermittent times, for example, during insect flight. And then, of course, we have to also contend with complexities across, for example, across species. So everything that we've spoken about today, I've been primarily focusing on the model organism of the vinegar fly, but you have to understand that different species have different ways in which they arrange their sensilla, and even the shapes of their antennae and the receptors which they have as players to actually receive outside signals. There's, of course, also age comes into play as a very big thing. So as you can imagine, many insects actually, especially holometabolous insects, they have to remodel from larvae as they come out of the egg, and they have to completely remodel their entire sensory organs during metamorphosis into, for example, these antennae that we've spoken about today. And we have to also remember that the repertoires are not the same during the larval stages of the life and the imago or adult stages of the life. There's also internal state, which is very important, which can, of course, modulate the way in which the cells and the receptors themselves work. So this can be, for example, things such as how hungry the insect is, if it has been mated recently, if it's a male or a female, of course. And for example, even with things such as whether if it's flying or walking, it can. There have been very many studies to show that insects can perceive things one way while they're flying and completely different ways while they're walking, and while they're hungry or while they're not hungry, and the like. And of course, there's also the complications and the complexity of actually dealing with the neurobiology of the insect, in which allows it to learn behaviors and to also have elicited behaviors, for example, socially. So the life history and the social context of the creature is also a very important consideration and an additional complexity that we have to wrestle with when trying to understand these little creatures. So there are, of course, a lot of different things, open questions still in the field that are very complex and difficult to actually answer, but we are making strides to actually try to understand them a little bit better. So here in this slide, I'll just go through a couple interesting questions that we still haven't figured out and that still remain a mystery, even though we've actually very well structurally characterized these sensilla and the receptors and the neurons themselves. So how do single activated receptors actually propagate signals, these initial, perhaps single receptor events, into entire action potentials that then fire to the brain? This remains an open question. We also don't understand very well how the OR complexes with its co-receptors and with other accessory proteins, and perhaps how it heterodimerizes or homodimerizes, and the exact structural biology of how the actual pores form and which receptors might combine with which other receptors in order to transduce the first currents into the cells. We also have some hints that there is neuron-neuron influence at the dendritic level, which we still very poorly understand. This is, of course, the case when there are more than one neuron innervating a single sensillum. We also don't really understand whether there exists a difference between neurons of a similar class. Even if they express the same receptor, we still don't know what other differences there may be with these neurons. Are they all created equal, or do they actually have some kind of hidden heterogeneity? We also don't really understand the role of olfactory binding proteins. So every few years there seems to be a paper coming out that shows that they are important and then falsifies that finding. And we've yet to really understand exactly in which cases and in which scenarios they are indispensable. I should mention that olfactory binding proteins are the most highly expressed transcripts in the antenna, and yet we understand very little about them, and the field is only growing. We also don't really understand exactly how receptors are trafficked all the way from the soma into these outer dendrites that you can see here, how they're expressed, how they're recycled. Do they get internalized and endocytosed upon binding, or do they just release their ligands freely? How this is maintained and how this is regulated throughout the life cycle of the animal, a very poorly understood question. We also have a very poor understanding of the functional role of all these non-neuronal cells, these non-red and pink cells. What are they actually doing? Are they dispensable? To what extent are they really actively contributing in the transduction process, and perhaps are they indirect players in these systems? And of course, we also have another question. Why the differences in the morphology of the sensilla? So as I've mentioned, there are club-shaped basiconic sensilla, for instance, and also these hair-like trichoids. Why do they exist in this configuration? What makes them different? And what are the evolutionary pressures that actually maintain this for so long in these insect lineages? I'd like to just mention as a quick aside, this is a neuron, and in our lab we have studied, for example, the ability for these neurons to actually propagate very, very minor signals that come in through these receptors into a train of action potentials that later actually goes to the brain. And we've just barely scratched the surface on how these how the sensitization process occurs. And just without going into much detail, I would just want to mention that even subcellular structures such as mitochondria play an active role in being able to determine what kinds of states a neuron might be in to receive signals. I think this is a very fascinating prospect, and I think a lot to still be studied. So finally, I'd like to just briefly explain to you how we as scientists, how we as biologists explore the antenna, and maybe share with you the methodologies that we have available to look into the antenna to better appreciate all these different levels of biological complexity and to answer questions about the ecology and the behavior and the neuroscience of these insects. I've broken it down into 5 basic subsections. Of course, by no means is this exhaustive, but for example, one way we have at looking at the antenna is through electrophysiology. So this involves us taking an insect such as the Drosophila melanogaster fly and performing electroantennography on them, which basically means we are measuring electrical currents from the antenna as they exist upon given stimulations of given odorants. So in the case of Drosophila melanogaster, one of the most infamous examples is the technique of a single sensillum recording, where we can actually insert electrodes into a very, very specific sensillum, not the whole antenna, but a very specific sensillum, and measure the responses from a specific sensillum one by one. And these recordings allow us to determine what each of the neurons are doing individually based on their amplitude, because the neurons don't fire with the same amplitudes on these recordings. And of course, while we are recording, we can present the flies with different stimulations. So for example, we can couple this with gas chromatography, and we can take, for example, a source of odorants, such as an apple, or perhaps the body wash of a female fly, and separate it into its constituent odors in such a way that we can present them one by one and study which sensillum and which neurons within which sensillum respond to which component of the mixtures. And this allows us to actually create a very nice map of which sensilla are responsible for detecting which odorants, and allows us to better paint a picture of the tuning curves of all the receptors. So the technique itself is very simple. We just extend out the antenna and we poke it with electrodes, and we get these kinds of measurements back. And as you can see, we can find very specific neurons that respond to specific compounds. It's a very, very powerful technique and allows us to study different kinds of flies, and we can present them with virtually any odorants in any order to study the responses. Of course, there are also techniques, electrophysiological techniques, such as open antennal preparations, where you take the fly's antennae and you cut them in half, and you make sure that the flies are expressing a genetically encoded indicator for calcium, so that when the neurons fire and the calcium levels transiently rise, you can see which cells are firing. And this is kind of the same principle as single sensillum recording, except that you don't really focus on the sensilla specifically, but you actually look at everything within a solution. So you don't puff gases and puff air on the sensilla that which you hope to record. You have everything under solution, and you look at the exact way the neurons themselves behave upon stimulation. So with this kind of technique, you can study sensitization and adaptation events, and also you can modulate in real time the processes of this neuronal firing by applying pharmacological treatments at the same time in order to block channels or keep channels open and the like. We also, of course, have a variety of microscopic techniques. The best one, I guess, for looking at the fly is electron microscopy. So in this case, you can see, for example, a scanning electron micrograph, where we can actually see the different kinds of sensilla. And we can also even look down to the very, very small ultra level scale, where we can see individual pores and how they are arranged and how they develop within each of these sensillas. So this electron micrograph very clearly shows how the pores look like through which the odorants can actually access the lymph and then arrive finely at the receptors on the neuron. We can of course also do transmission micrographs, where we can actually study the exact cellular structure beneath, and this has allowed us to get a better understanding and paint a better picture of exactly how the sensillum looks like and how the biology underneath it all looks. We also have very many techniques that we apply from molecular biology and from the neurogenetics toolkits. One that I'd like to mention briefly that we've used for identifying which cells express which chemosensory proteins and odor and binding proteins. This is of course the technique of fluorescent in situ hybridization, where we can tag genomic transcripts fluorescently to study which cells are expressing which genes. And this has been quite fruitful in understanding that not all odor and binding proteins are present everywhere, but that they're actually very carefully and stereotypically segregated into specific sensilla where they actually perform their function. We also have the techniques, for example, of transcriptomics, where we can compare or where we can exploit the fact that we have a very deep literature and understanding of mutations in flies. And we can compare, for example, the transcript levels between different mutants and see what kind of mutations have which kinds of effects on the actual transcriptional landscape in these animals. And of course, with the advent of single cell sequencing, we might also one day be able to look into the antenna on the single cell resolution and actually understand which cells express which genes and better understand how the things are partitioned and what the real heterogeneity of the antenna really truly is. We also have a very infamous technique called the Gal4 Uas system that we can use in flies. Very briefly stated, you take two elements from yeast and you can introduce them into the chromosomes of flies. One of the elements is a Gal4 element, and another is a Uas element. And basically what you can do is you can fuse these to the promoters or enhancers of genes in the case of Gal4, and you can fuse the other element, the Uas element, to reporter genes, whichever reporter gene you like. And by bringing these two things together, you can achieve a rather simple but very useful thing of being able to express your reporter gene in a very specific line of cells. So for example, we could take a Gal4 line for the olfactory sensory neurons. So we could specify where we want our reporter gene in the olfactory sensory neurons, and then with the use of the Uas, we could actually make these cells glow green with the help of a fluorescent marker, for example. So with this kind of technique, you can interfere with the expression in the cell. You can mark the cells. You can remove them. You can overexpress things and the like. And one of the most prolific uses of this technique is called the empty neuron system, which has been exploited very frequently in the field, where you take the case of one sensillum, which expresses one receptor, and you mutate it and knock out this receptor so that you actually find yourself with an empty neuron. And then by expressing different olfactory receptors in this neuron, you can study the receptor itself in a completely ectopic and foreign environment. And this has been used very frequently in order to atlas different receptors and what different receptors do. And you can even take receptors from different insects, for example, hawk moths or mosquitoes, and you can actually bring them into the fly empty neuron system. So you can take a fly and study receptors from all different kinds of insects with this technique. And this is reliant on the Uas Gal4 system. There are, of course, heterologous expression studies where we can also use completely different cell lines and express our receptors or olfactory binding proteins, odor and binding proteins. So to date, we typically have used mammalian cell lines and insect cell lines and xenopus oocytes in order to transfect them with receptors and study what kind of kinetics and responses they have outside of the original system. And of course, in the lab, going beyond the antenna, we have a lot of experiments that look at the complexities of the brain, and of course, eventually the actual resultant output behaviors that come from the insects interacting with the outside world. So these behavior experiments are beyond the scope of this talk, but I'll very quickly mention that we employ choice assays and mazes and traps in order to figure out which smells an insect might prefer, and that we can also mutate different genes and see whether these trends still hold, or whether perhaps some genes might be important for the fly's ability to make some specific decisions. And then, of course, eventually, one recent development in the field was to actually make an entire virtual reality for the fly, where you can control the stimuli that the fly receives on many fronts, on a multimodal front. So, for example, you can puff odorants in a very controlled fashion and also control what the fly sees, and then track its movement throughout the system. So for this section, I'd like to just mention that one very major goal that we still continue to pursue is the atlasing of the antenna and its mechanisms. So we always ask questions such as, where are the sensilla positioned on the antenna? Which receptors are found in which sensilla? Which odors does each receptor respond to, whether or not, whether it is a widely tuned receptor that responds to many odorants or not? Which accessory proteins modulate the responses to which odorants and in which sensilla they're found in? And of course, what are the differences in antennal organization between species, which I haven't touched upon very much. But as you can imagine, antennae look different, so they might be organized in a very different way, and perhaps even gain additional features, which might be dependent on being a larger shape as opposed to a very bulbous kind of vinegar fly antenna. I will not get into that for now. So now we've completed our 5 chapters. We've explained why antennae exist, what they're equipped with, how everything is structured together, and all the different levels of biological complexity that arise from this organization. And of course, I've given you a little bit of a primer on what kind of things we can do in the lab to even look into this very tiny appendage of the insect. So that brings me to the end. I'd like to give you a very short 5-sentence summary of the whole talk. So antennae are nature's invention to tackle a turbulent terrestrial world of chemical signals, as I described in the beginning. These antennae are equipped with a repertoire, and this repertoire includes secreted proteins and specialized receptors that perform the actual transducing functions. The antenna is a compartmentalized organ, a very heavily isolated and compartmentalized organ composed of very many hairs called sensilla, which create these distinctive chemosensory units, and these and that this whole sensory process is functionally regulated at many levels of complexity and at many scales of complexity, many of which are unexplored. And of course, this leaves the field with insect antennae as a very fruitful subject for research and application at the crossroads of behavior and ecology and the very deep study of neurobiology. So with that, I'd like to acknowledge my department and thank everybody who has helped me present all of this. I'd like to give a special shout out and thank you to Bill Hansen, who is the head of the department, and to my supervisors Sophia and Dieter. Thank you very much for your guidance. And of course, I thank everybody else, too many to name, who have helped me actually prepare this talk and try to give you a rather brief, although I know it went on a little bit, overview of everything that there is to say about insect antennae. With that, I'd like to thank you for your attention, and yes, have a good day. Thank you very much.